Hello viewers, and once again welcome to my channel. My name is Delvin Wenani, and today I want to take you through KCSE Biology Paper 2, KCSE Biology Paper 2, the year 2022, Section B. Now, Section B contains 40 marks. Section B contained 40 marks, and out of the 40 marks, we have 20 marks for question number 6, and then question number 7 and 8, each one of them also has 20 marks, but you pick either of the two. So you pick one of the 7 and 8. So question number 6 has 20 marks, which is a graphical question, and then between question number 7 and 8, you pick one. So when you pick one, which also contains 20 marks, totaling to 40 marks. So section B has 40 marks. So ladies and gentlemen, remember to subscribe to my channel, remember to follow, remember to share so that we can reach as many people as possible, and don't forget to comment so that where we can be able to help, we can be able to help. So let us reach the comment section so that we can be able to grow together. So ladies and gentlemen, in the year 2022, question number 6, paper 2 of biology was, and I read, a shoot of an aquatic plant was exposed to different light intensity and the rate of photosynthesis estimated by counting the number of bubbles of a gas leaving the shoot per minute. The results were tabulated as shown below. So the results we have here, we have the results, the number of bubbles per minute, and then we have the light intensity. So number of bubbles per minute ranging from 0 up to 39. Then the light intensity from 0 to 8. Now, on the grid below, that's question number A, draw the graph of the number of bubbles produced per minute against the light intensity. So we are drawing it against light intensity. Total marks, 6 marks for plotting that graph. Now, the first thing we are supposed to do is we look at the space that we have. That's the space that we have been provided with. So we shall manage that space, whereby it's recommended that at least we use 3/4 of that page. So looking at the page we have, we now go back to our values, and the values we have the number of bubbles per minute ranging from 0 to 32. Now remember, this one shall be on the y-axis. Light intensity shall be on the x-axis. So this one shall be on the x-axis. On the x-axis. Then, all said and done, looking at also the graph that we have, we can also give our scale, and the scale on the y-axis, looking at the space that we have, the scale on the y-axis, let us start with the y-axis. Remember, the y-axis is ranging from 0 to 32. So the axis that is the scale, the scales we're going to use. On the y-axis, we shall have 1 centimeter to represent 5 units. Keeping in mind what we have. Then on the x-axis, we shall have 1 centimeter representing 1 unit. So those are the scales we're going to use. Y, we're going to use 1 centimeter to represent 5 units. Then on the x, we're going to use 1 centimeter to represent 1 unit. Now, let us go to our graph now. So the first thing is I'll be able to plot to draw the x-axis. This is the x-axis. This is my x-axis. Then this is my y-axis. Now, then we label the axis. On the x-axis, we are being told that we have light intensity. So on this side, we have light intensity. Now we have said we are going to use a scale of 1 centimeter to represent 1 unit. So we shall have our origin here. Our origin here as 0. So we shall have our origin here as 0. Then 1, 2, 3, 4, 5, 6, 7, 8, and then 9. Remember, our value ranges from 0 to 8, so 9 is okay. Then on the y-axis, we shall also have another origin on the y-axis. That is, we're going to use another 0 here. Then we shall go with 5 units, 5. Then we have 10. We have 15. We have 20. This is 20. Then we have 25. Then we have 30. And then we have 35. Remember, the highest unit on the y-axis is 32 units. And also to label the axis, on the y-axis, it is the number of bubbles per minute. So on this side, number of bubbles per minute. So that is it. So next, now we want to plot. Now we move to the plotting. We want to plot those points. So the first point, allow me to pick another color for plotting. The first point is when the number of bubbles, the first point is when the number of bubbles is 0, the light intensity is also 0. So that is the starting point, the origin, have 0, 0. Then when the light intensity is 1, it is 9. The number of bubbles is 9. So when it is 1, it is 9. When the number of bubbles, when the light intensity is 2, the number of bubbles per minute will be 16. Yeah. When the light intensity is 3, it is 22. This is 21. 22 is here. When it is 4, that is the light intensity, then this one is 28. 28, just above. This is 25, 26, 27, 28. When light intensity is 5, it is 31. 5, it is 31. When it is 6, when the light intensity is 6, it is 32. There's 32 here, above 31. 7 is also 32. And 8 is also 32. Good. So that is the plotting. Now, after plotting, now we join those points. We join the points of plotting. And when you're joining the points, kindly join with a smooth curve. As smooth as possible. So without, and it should be as thin as possible to increase the level of accuracy. So we join those points. Join the points. Like that. Those are the points joined together. Now, once you have done like that, then you will have secured 6 marks in that question. And this is how it's going to be marked. This is how the question shall be marked. Out of the 6 marks, the candidates shall score as follows. For good plotting, remember it's only one graph, so we shall mark plotting. Plotting, you'll get 1 mark. We also have labeling. If you have done very good labeling of the axis, that is labeling of the axis, you get 2 marks because there are 2 axes. If you have a smooth curve, if your curve is very smooth, so you have a smooth curve, you'll have 1 mark. Remember, it's only 1 curve. And if you have a scale, but the scales I talked about up there, the scale, then the scale shall give you 2 marks. This one shall total to, so this will total to an average of 6 marks. So you'll get 6 marks in that question. Remember also there are some do's and don'ts, and one of them is you make sure that your scale must be workable. That is, you use a scale that is workable. Also, make sure that when you're doing your plotting, you make sure that your points are seen very clearly so that you can get your marks, all of them. Remember, if your plotting is incorrect, then you miss the plotting mark and the curve. You are denied the plotting marks and the curve. You'll miss the 2. So, ladies and gentlemen, let us now move on to question number B. Question number 6B. State how the identity of the gas produced can be determined in the laboratory. State how the identity of the gas produced can be determined in the laboratory. Now, the gas being produced here, remember, it is oxygen because of photosynthesis. The process actually is photosynthesis, and therefore the gas being produced is oxygen. So what the procedure we're going to use in the lab is to test if oxygen is present. And the common procedures we use in the lab is using a glowing splint. And what happens is oxygen will relight the glowing splint. Now, the other question is name the apparatus used for measuring light intensity. Name the apparatus used for measuring light intensity. So the apparatus used in measuring light intensity, they're given so many names. You can use what we call a photometer. You can also use another one known as a lightimeter. We also have an exposure meter, photographic lightimeter. Any of those ones is correct. So those are some of the apparatus used. Now, why was it necessary to get the shoot from an aquatic plant? Why was it necessary to get a shoot from aquatic plant? Now, we understand that aquatic plants are adapted to photosynthesis underwater, or under what we're calling limited light intensity. And therefore, they can photosynthesize even when the intensity of light is too low. So they utilize the CO2 in solution form that is dissolved in water to produce gases, that is oxygen, from the aquatic plants underwater. Now, the other question is account for the number of bubbles produced between the following units of light intensity. Now, between 0 to 6. Between 0 to 6. And remember, it is 3 marks. First of all, when you look at the graph between 0 to 6, there's what we call a rapid increase in number of bubbles produced. And this is because of what? This is because of an increased light intensity, which increases the rate of photosynthesis, Now, that's from number 2, we are also counting between 6 to 8. Between 6 to 8, the number of bubbles produced remain constant since the rate of photosynthesis had reached what we call the maximum. And this is also due to what we call the optimum light intensity. Question number F, state 2 modification one will make on the experimental setup to increase the rate of gas bubble production. Now, number one is to supplying of sodium hydrogen carbonate in the setup. Number two is also to increase in number of shoots in the setup, and also to increase in temperature. Question number G, explain the limitations of using gas bubbles to determine the rate of photosynthesis. What are some of the limitations? We understand that photosynthesis and respiration occur simultaneously, and therefore, if you are trying to use photosynthesis to determine the amount of gas produced, remember also we have respiration taking place at that time, and respiration is consuming the oxygen being produced. So not all the bubbles may be accounted for. Some gas will dissolve in water also. Question number H, with a reason, predict the number of bubbles that will have been produced at 15 units of light intensity. That is light beyond, way beyond our graph, because our graph was reaching 8. So it will be 32 or any other value less than 32. And this is because photosynthesis had reached the peak. Question number 7. Question number 7 is an essay question. And in this question, we are being asked, if I read through the question is asking us to describe how plants eliminate waste products. That is 8 marks. To describe how plants eliminate waste products. 8 marks. So in this question, remember it's an essay kind of a question, and we are being asked to describe how plants eliminate waste products. So I'll suggest that we describe each way, because we are being asked on ways on which plants eliminate waste. So I'll be suggesting a way and also giving an explanation. Suggest a way and then give an explanation for question number 7A. The first one is through transpiration. And here I'm talking about excess water, which is lost in form of water vapor through the stomatas and also through the lenticel. That's number one. Number two is also through what we call diffusion, or what we call recycle or reuse. Now we understand carbon dioxide, which is produced from respiration, and oxygen, which is produced from photosynthesis, as waste products, are eliminated through lenticels or cuticle or the stomatas. But the same gases, that is the oxygen and carbon dioxide, are reused in respiration and photosynthesis respectively. So they are being reused at the same time. Number three is through deposition and storage, what we call storage in non-toxic forms. That is also what we call the organ fall. Now, some waste, e.g. what we call the calcium oxalate, we also have caffeine, nicotine, quinine. Majority of these waste are stored in non-toxic forms in tissues like leaves, flowers, bird seeds, fruits, and are eliminated through the fall off of these some of these tissues like the leaves. We also have exudation. These some waste are excreted in semi-solid forms, in form of salts, that's the latex, gum, calcium, pectate, and also resins. We also have guttation. Number 5 is guttation. Now, guttation is whereby a plant lose excess water in form of water droplets through what we call the hydrothoids, Now, with one, we're giving a maximum of 8 marks. That is for a method and an explanation, a maximum of 8 marks. Question number B is at 7B. Describe the structure and function of the mammalian nephron. Describe the structure and function of the mammalian nephron. it has a renal tubule. It also has a glomerulus, which has a rich network of blood capillaries. We can also see that it has a Bowman's capsule for passage of glomerular filtrate into the proximal convoluted tubule. And it also has a glomerulus where ultrafiltration occurs. And this is because of due to the difference in diameter of the blood vessels having it. It also has, if you check, just check kindly, you see it has afferent blood vessels, what you call the arteriole. And also it has efferent, that is blood vessels, or what you call the arteriole. It also has the proximal convoluted tubule. It has the proximal convoluted tubule, which extends from the Bowman's capsule. And you can see it extending from the Bowman's capsule, whereby the filtrate flows through the proximal convoluted tubule, where useful substances such as glucose, amino acids, some water, and mineral salts are selectively reabsorbed into the blood stream by a process known as the active transport, diffusion, or osmosis. It also has the loop of Henle, or sometimes we call it the Henle's loop, which is in form of ascending and descending. Now, the descending loop of Henle is for reabsorption of water, while the ascending loop of Henle is for reabsorption of mineral salts. Now, look at that diagram. You'll see it also has a distal convoluted tubule, and this is where much water is reabsorbed into the bloodstream by the process known as osmosis under the influence of a hormone known as ADH. And more mineral salts are reabsorbed by diffusion and sometimes active transport under influence of aldosterone hormone. It also has a collecting tubule. It has a collecting tubule for reabsorption of more water. Collecting tubule at the end for reabsorption of more water. Question number 8. Describe 5 tropic. Question number 8A. Describe 5 tropic responses in plants and their survival values. Describe 5 tropic responses in plants and their survival values. Now, we shall go straight to those responses and their survival values. Number 1 is phototropism. Phototropism. And maybe the definition for phototropism, this is a growth or a curvature, the growth of a curvature in response to direction of light. We call it phototropism. Growth curvature in response to direction of light. And you can see from that photo how the growth is, how the curvature has grown towards light. That is the shoot. Now, the effects on the shoots and the roots are, we have what we call shoots are positively phototropic, while roots are negatively phototropic. And what is the survival value for this? The survival value is to reach light for photosynthesis, to reach light for photosynthesis. Number 2 is chemotropism. Chemotropism. Now, this one is a growth response to chemical or substance or minerals or ions. And this growth is usually, an example is the growth of pollen tube towards the embryo sac, and also the growth of roots towards mineral salts. And this one is for absorption and also for uptake. Number 3 is what we call geotropism or gravitropism. This one is a growth response to gravity. This is whereby the shoots are negatively geotropic, while the roots are positively geotropic. Now, this one enables the survival value of this. It enables the roots to reach water and mineral salts for absorption, and also enables the roots to grow into the soil and offers the plant what we call anchorage or support. Another response is what we call hydrotropism. Now, this one is the growth response to water or moisture. Here is whereby now the roots are positively hydrotropic, and it enables the roots to reach water for absorption. That is a survival value. We have another one known as the Number 5 is thigmotropism, or what we call haptotropism. Now, this is a growth response due to contact with solid objects, and this is whereby now it is observed mainly in climbing stems, where these plants which employ the use of tendrils, and especially in weak plants, plants which have weak stems, to enable them to reach light, and also for photosynthesis, and also for to enable their flowers to be exposed to pollinators, and also expose fruits for dispersal. Those are the survival values. Question number 8b, which states, describe how the mammalian heart is controlled. Describe how the mammalian heart is controlled. Now, I want to say this. The mammalian heart is controlled in 2 ways. And with the first way is what we call during the non-nervous control. Remember, the cardiac muscles are myogenic. That is, they initiate their own contractions and relaxations. And also, stimulations and excitements are all originating from a point on the heart, which we call the SAN, that is the sinoatrial node. And this one stimulates or produces excitations through charges which spread through auricles, leading to these contractions. Now, stimulations will proceed to the atrioventricular. The stimulations will proceed to atrioventricular node, AVN, into what we call the pancreal tissues, to all ventricles which contract. To all ventricles which contract. Now, there's also during the nervous control. During the nervous control, how does the heart function? Now, this one now comes whereby when the blood pressure and the chemical composition of blood is monitored by cardiovascular center of the brain, the rate can be increased by what we call sympathetic nerve. That is a heartbeat rate lowered by vagus nerve. During the nervous control, blood pressure and chemical composition of the blood is monitored by what we call the cardiovascular center of the brain. So the rate can be increased by sympathetic nerve, and heartbeat rate can also be lowered by what we call the nerve, the vagus nerve, by what we call the vagus nerve. The heartbeat rate can also be lowered by the vagus nerve. So ladies and gentlemen, with that one comes to the end of this paper. Remember to share, remember to follow, remember to subscribe, and remember to share to your friends so that we can reach as many people as possible. But ladies and gentlemen, also don't forget to subscribe so that the next time I post the next video, you'll be notified. Otherwise, I want to take this opportunity and appreciate you very much for your time. May God bless you and have a nice time. Thank you very much. Thank you very much.